Welcome to this explainer. Today we are exploring one of the most fascinating and honestly high-stakes mechanisms in all of human biology. Think of your immune system like a ridiculously high-performance sports car. To navigate the really treacherous roads of viruses, bacteria, and malignancies, it needs a massive engine and a powerful accelerator to just eliminate those threats quickly. But just as importantly, it needs highly responsive, precision-engineered brakes, because without them, that immense power would just cause the car to crash, totally destroying the very host it's meant to protect.
Okay, let's dive right into this core dilemma. If the immune system is overly aggressive, it fails to recognize its own tissues, right? And that leads to severe autoimmune diseases where host organs are literally destroyed by friendly fire. But on the flip side, if the immune system is too weak, or if its brakes get stuck in the on position, the body just easily succumbs to chronic viral infections or cancer. It is absolutely the deadliest balancing act in biology, and understanding how it works is completely unlocking the future of medicine.
So here is our roadmap for today's explainer. 1. The Immune Balancing Act. 2. Mechanisms of immune tolerance. 3. The threat of T-cell exhaustion. 4. Checkpoint inhibition cancer therapy. 5. Engineering CAR T-cells. And finally, 6. The aging immune system. All right, let's kick things off with sections 1 and 2, the Immune Balancing Act and the mechanisms of immune tolerance. This whole balancing act is really all about those biological brakes designed to prevent self-destruction. And the master key to these brakes is a concept called tolerance. By definition, tolerance is a state of non-responsiveness in our T cells and B cells. Basically, when these cells encounter antigens, whether they're foreign invaders or just from our own tissues, but they don't get a secondary danger signal from co-stimulatory molecules, they just stand down. This critical non-responsiveness is what stops our immune cells from unleashing their destructive potential on perfectly healthy cells.
Now, our bodies maintain this tolerance through two main security systems. First, there's central tolerance. This happens super early on in the primary lymphoid organs, so the thymus for T cells and the bone marrow for B cells, through this incredibly ruthless process called negative selection. If newly minted immune cells react too strongly to self-antigens, boom, they are simply deleted. But, some rogue autoreactive cells always manage to escape. And that's where peripheral tolerance comes in as a vital backup system. Operating out in the lymph nodes and spleen, it catches these escaped cells and neutralizes them through active suppression or a process known as anergy.
And anergia is a truly fascinating state. Actually, think of it like putting a highly reactive toddler in a permanent timeout. These dangerous immune cells aren't killed off. Instead, when they encounter a self-antigen without those required co-stimulatory signals we talked about, their internal wiring is just flipped into an unresponsive, inactivated state. So they remain alive, just circulating through your body, but they are functionally neutralized. They're completely unable to trigger inflammation. And this brilliantly illustrates how the active suppression side of peripheral tolerance works. It relies heavily on a specialized group of cells known as regulatory T cells, or TREGs for short. Driven by a really crucial transcription factor called FOXP3, these CD4 or CD8 T cells are the dedicated peacekeepers of the immune system. They patrol the body and secrete inhibitory cytokines, things like interleukin-10 and TGF-beta. So when regular T cells try to start an unwarranted fight, these TREGs step in and suppress the response, keeping the tissue totally harmonious. To really understand how vital these peacekeepers are, we just have to look at what happens when they fail. Monogenetic mutations, so literally just errors in a single gene, can cause devastating immune dysregulation. If the FOXP gene mutates, regulatory T cells just can't develop properly. That leads to a severe condition called IPEX syndrome, which is characterized by massive systemic autoimmunity. Similarly, mutations in the CTLA4 gene severely decrease TREG function, resulting in multiple super dangerous autoimmune syndromes. Without the brakes, the car absolutely crashes. But what happens when the immune system faces an enemy it simply cannot clear? That brings us to section 3, the threat of T-cell exhaustion. T-cell exhaustion is essentially the immune system falling asleep at the wheel. It occurs when T cells are subjected to constant, unending stimulation by persistent antigens. The memory function of the cell gets completely disrupted. We see this most frequently in chronic viral infections like HIV-1 and hepatitis C, and quite prominently in cancer. The T cells are there. They completely recognize the enemy, but they are just too burnt out to fight. We can actually identify these exhausted cells by their biological signatures. They're like overworked soldiers. Because of that persistent antigen exposure, they begin expressing really high levels of inhibitory receptors like PD-1 and CTLA4 right on their surface. As exhaustion sets in, their ability to proliferate is deeply compromised. Ultimately, they face a total loss of cellular function, basically losing the capacity to produce the vital attack cytokines they desperately need to kill target cells, stuff like interleukin-2, TNF-alpha, and interferon gamma.
Moving right along to Section 4, checkpoint inhibition cancer therapy. For decades, tumors exploited this exact exhaustion mechanism to survive. So the crucial point is understanding exactly how the tumors force our T cells into exhaustion in the first place. Under normal circumstances, for a T cell to activate, a molecule on its surface called CD28 needs to bind to B7 ligand on an antigen-presenting cell. But CTLA4, which is expressed on that exhausted T cell, has a much higher affinity for those B7 ligands. It physically outcompetes CD28, binds right to the B7, and transmits a powerful inhibitory signal. It acts as a massive off switch. Tumors also upregulate ligands for another off switch, PD1. By pulling these specific biological levers, cancer cleverly hides in plain sight. But recognizing this, scientists developed a profound counterattack. The mechanism works in a few sequential steps. First, we identify that the tumor is exploiting the CTLA4 or PD1 pathways to induce that exhaustion. Second, we administer specialized drugs called monoclonal antibodies, which are specifically designed to target these very pathways. Third, these antibodies bind directly to CTLA4 or PD1, physically blocking those inhibitory receptors. In essence, they are quite literally cutting the brakes. Finally, free from that suppression, the exhausted T cells are violently reawakened. Their exhaustion is totally reversed, and they are unleashed to recognize and destroy the tumor cells. The result is this beautiful, explosive reactivation of the immune system. And this isn't just theory anymore. These checkpoint inhibitors, drugs like ipilimumab, nivolumab, and tremelimumab, are fully FDA approved right now. By simply taking the brakes off the patient's own immune system, doctors are successfully shrinking solid tumors in melanoma, kidney, lung, liver, and head and neck cancers. It's an absolute game changer. Which leads us perfectly into Section 5, engineering CAR T cells.
But what if taking the brakes off just isn't enough? What if we need to completely rebuild the engine? Well, CAR T cell therapy involves taking a patient's own T cells and giving them a massive synthetic upgrade. Using recombinant techniques, scientists design a chimeric antigen receptor, or CAR. It combines an extracellular targeting domain, which is essentially an antibody fragment that acts as a highly specific radar for cancer cells, with powerful intracellular signaling domains taken from co-stimulatory molecules like CD28 or 41BB. When we put this hybrid receptor back to the patient's T cells, we create an unstoppable supercell, perfectly retargeted to hunt down and attack specific malignant cells, particularly in leukemias and lymphomas.
Let's move to and see how this builds into our final topic, Section 6, the aging immune system.
All of this incredible science eventually runs into a challenge every single one of us inevitably faces. As we get older, our biological sports car, well, it inevitably gathers some rust. And this results in a really fascinating paradox. On one hand, we see a broad decline in both innate and adaptive immunity, meaning older individuals just have a much harder time fighting off new pathogens or generating strong responses to vaccines. But simultaneously, there is an immune overactivity. The aging immune system produces a paradoxical increase in baseline pro-inflammatory cytokines. So think about it. It's weaker at fighting the enemies it should fight, but it drives chronic inflammation and disease against the host. Yet, despite the challenges of aging and disease, our mastery over these immune mechanisms has literally never been greater. As the source material emphatically notes, manipulating the immune system's regulation, specifically through immune checkpoint therapy, has now officially joined surgery, radiation, and chemotherapy as a primary pillar of human medicine. We are no longer just poisoning tumors, we are fundamentally reprogramming the body to cure itself. And that leaves us with a truly inspiring question to end this explainer. We now know how to install synthetic targeting radars. We know exactly how to cut the molecular brakes. And we understand the incredibly delicate balance of immune tolerance. So, as we learn to expertly control the immune system's accelerator and brakes, what currently incurable disease are we going to conquer next? The microscopic battles happening inside you right now truly hold the key to the future of human health. Thanks so much for joining me, and keep learning.